Hello, I'm Professor Sims, and in this video I will discuss the respiratory system. This is the 6th in a series of 10 lessons held as part of my Anatomy and Physiology 2 online laboratory course. If you're a student currently enrolled in this course, please consult the syllabus and course Moodle site for assignments, quizzes, due dates, and other course information. By the end of this lesson, you'll be able to identify and describe the structure of the upper and lower respiratory system, describe the mechanism of carbon dioxide and oxygen exchange, define the size, shape, and location of the lungs, understand the physiology of inspiration and expiration, and we will also describe the significance of respiratory volumes and capacities. Let's begin with an overview of the respiratory system. So the primary function of the respiratory system is to enable gas exchange. So what that means is you're bringing oxygen into the body and you're removing carbon dioxide, which is a waste product of cellular metabolism. And this exchange is vital for maintaining proper blood pH and for supporting energy production in all of your cells. As air enters the body, it travels through a series of structures that filter, warm, and humidify the air before it reaches the lungs. This conditioning process protects the delicate tissues in the lower respiratory tract and helps to optimize gas exchange. In this diagram here, you can see the major structures labeled, including the nasal and oral cavities, the pharynx, larynx, trachea, right and left main bronchi, and the lungs. And also shown is the diaphragm, which is a critical muscle that drives ventilation by contracting and relaxing to move air in and out of the lungs. And then at the very end of the pathway, inside of the lungs, air reaches the alveoli, which are tiny air sacs, where the actual gas exchange occurs across thin capillary walls. The system is anatomically divided into two main parts: the upper respiratory tract, which includes the nose, the pharynx and larynx, and then the lower respiratory tract, which includes the trachea, bronchi, and then the lungs. Let's take a closer look. This part of the respiratory tract, the upper airway, includes the nasal cavity, the pharynx, and the larynx. And then together, these regions serve to condition and direct incoming air toward the lower respiratory system. So as the air moves through the nose or mouth, it passes through the nasal cavity, where it's filtered by hair and mucus, warmed by blood vessels, and then humidified in order to prevent irritation in the lung. This process ensures that air reaching the lungs is clean and at body temperature, which is important for protecting the delicate alveolar surfaces. Then from there, the air moves through the pharynx, a shared pathway for both air and food, and then it enters the larynx, also known as the voice box, which plays a dual role in both airway protection and sound production. A key feature here is the epiglottis. We've seen this before. It's a flap-like structure that prevents food or liquid from entering the trachea during swallowing. Nearby, the vocal cords down here, or the vocal folds, vibrate as air passes through, allowing us to produce sound. And altogether, the upper respiratory system not only prepares air for gas exchange, but also contributes to important functions like speech, swallowing, and of course, immune defense. This here is just a closer look at the structures of the larynx and the vocal cords. Again, here up at the top is the epiglottis. Then you have thyroid cartilage. We've seen this before. A hyoid bone, that should also be familiar to you guys at this point. And then this part here is part of the tracheal cartilage. So that's part of what helps to move and warm the air. And then this is a close-up of the vocal cords, the vocal folds, and it's showing the vocal cords in relation to the epiglottis and the esophagus and the trachea here. Now that was the larynx. That's the larynx and vocal cords. Now let's look a little closer at the pharynx. The pharynx is divided into 3 distinct regions, and they all have a specific function and location. So first we have the nasopharynx, the uppermost portion, which is located directly behind the nasal cavity. This section functions strictly as an airway, and it connects to the auditory or eustachian tubes, which help to equalize pressure between the middle ear and the atmosphere. You'll often feel this pressure shift when you change altitude, like during a flight in an airplane. If you move downward from the nasopharynx, we enter the oropharynx, which is located behind the oral cavity. This region serves as a shared passage for both air and food. And then because of this dual role, the tissues lining the oropharynx are more resistant to abrasion than those in the nasopharynx. And then the laryngopharynx. The laryngopharynx is the lowest part of the pharynx, and it opens into the larynx, which leads to the trachea and lungs, and also the esophagus, which leads to the stomach. And this area is responsible for directing air into the respiratory tract and food into the digestive tract, which makes it a really important key junction in the upper airway. This figure is showing more details about the trachea itself, and again, it's helping you to kind of orient where you are as far as larynx. This is below where we were just looking at. So it's pharynx, larynx, and then trachea. And then down here, we're looking at the bronchi in the lungs. This here is a histology slide showing the location of the lumen, cilia, submucosal glands, and hyaline cartilage of the trachea. If we're looking at the respiratory zone, this is the portion of the respiratory system where the actual gas exchange occurs. So we're talking about bronchi, bronchioles, alveoli, all of those kinds of things. So this is beginning with the bronchioles. The bronchioles then branch into alveolar ducts and then terminate in clusters of alveoli. And alveoli are the tiny air sacs that are the functional units of the lungs. Unlike the conducting zone, which includes structures like the trachea and bronchi, the respiratory zone lacks cartilage. It doesn't have goblet cells, and these structural differences are important because this allows for the maximum surface area and minimal barrier thickness, which are important for efficient gas diffusion across membranes. And the most critical components here are the alveoli. They are microscopic sacs with extremely thin walls, closely associated with capillary networks. Oxygen from the air diffuses across the alveolar membrane into the blood, while carbon dioxide diffuses out of the blood and into the alveoli to be exhaled. So altogether, the respiratory zone is where the lungs do their most important work, facilitating the exchange of gases that keep your body alive and functioning. This is a closer look at the alveoli. You can see the alveolus here. This is a gas-filled space, and you have macrophages, capillaries, alveolar pores. This is a histology slide showing some of these structures as well. This big empty space here is the alveolar sac. Then you have the lumen of the bronchiole and individual alveoli over here. Let's switch focus to the lungs. The lungs, of course, are the primary organs of the respiratory system. They are a pair of spongy, elastic organs located in the thoracic cavity on either side of the mediastinum. Structurally, the right lung is divided into 3 lobes, the superior, middle, and inferior lobes. And then the left lung only has 2 lobes. And the reason for this, well, first of all, you have the superior and inferior lobe. And the reason you only have 2 lobes here is so that there is room left for the heart. Each lung is enclosed in a pleural membrane, which has 2 layers. The visceral pleura that directly covers the lung surface, and the periantal pleura that lines the chest wall. And then between them is the pleural cavity, a thin fluid-filled space that reduces friction during breathing movements and helps keep the lungs inflated. Inside the lungs, the brachial tree branches into increasingly smaller airways, eventually terminating in the alveoli, which again are the tiny thin-walled structures where gas exchange takes place. And oxygen moves from the alveoli into the blood while carbon dioxide moves out to be exhaled. This slide helps to illustrate the mechanics of the pulmonary ventilation, which is just the movement of air in and out of the lungs. Breathing is driven by pressure changes in the thoracic cavity, which are created by muscle contractions and the elastic properties of the lungs. So at rest, the atmospheric pressure and intra-alveolar pressure are pretty much equal, typically around 760 mmHg, which means there's no net movement of air. Next, if we were to look at the intrapleural pressure, pressure. Intrapleural pressure is the pressure within the pleural cavity between the parietal and visceral pleural, and it is normally about 756 mmHg, which is slightly lower than alveolar pressure. And this negative pressure, known as transpulmonary pressure. Transpulmonary pressure is the difference between the alveolar and interpleural pressure, and it's important for keeping the lungs inflated. And in this example, we're talking about 4 mmHg, and the way you got that is from subtracting 756 here from 760. Now, during inspiration, the diaphragm contracts and flattens, and the external intercostal muscles contract, expanding the thoracic cavity. And then this expansion lowers the intra-alveolar pressure below the atmospheric pressure, which allows air to flow into the lungs. During expiration, the diaphragm and intercostal muscles relax, the thoracic cavity volume decreases, and the intra-alveolar pressure rises above the atmospheric pressure. As a result, air is pushed out of the lungs. So together, all of these pressure changes and muscular movements create the essential rhythm of breathing, which of course is bringing oxygen in and sending carbon dioxide out of the lungs. Spirometry, which is a simple and non-invasive test that measures how much air you can inhale, how much you exhale, and how quickly you can exhale. So this is the terminology. Let's go through that. Tidal volume, TV, is the amount of air you breathe in or out during a normal, quiet breath. This is your basic resting breathing pattern, about 500 milliliters in a healthy adult. Inspiratory reserve volume, IRV, refers to the extra volume of air that you can inhale after taking a normal inspiration. Imagine taking a deep breath in after a normal breath. That's your IRV. Expiratory reserve volume, ERV, is the additional air that you can forcefully exhale after a normal exhalation. This helps to assess whether your lungs can efficiently push air out. And then vital capacity, VC, is the total amount of air that you can move in and out of your lungs with one deep breath in, followed by one deep breath out. It combines total volume, inspiratory reserve, and expiratory reserve, and is often used to monitor conditions like asthma, COPD, or recovery from a respiratory illness. And by looking at these numbers, clinicians can detect lung disease, track progression, or evaluate treatment success. For the Lab 6 pre-lab activities, you want to make sure you read the Respiratory System chapter. This is chapter 22 in the OpenStax book. Review the introduction and background sections, as usual, of the lab manual. You want to do the pre-lab exercises and use those answers to help you complete the quiz on Moodle. For lab exercise 6.1, you will use the models to identify the diaphragm, epiglottis, thyroid cartilage, thyroid gland, trachea, right lung, left lung, hyoid bone, right primary bronchus, and left primary bronchus. So these are some examples, of course, of the anatomical models that you would see if you're in a face-to-face lab. For my online students, you could just use the figures that are in the lab manual. But these are the larynx, the external structures here, internal structures here. And then I'm showing this model along with this figure here so that you can see the left primary bronchus, left lung, these are over here. This big large bit down at the bottom, that's the diaphragm. This is the right lung over here, and of course your trachea, esophagus. It's hard to see here, but again, you got your hyoid bone, your epiglottis. Those things can also be seen there. For exercise 6.2, you're going to use the histology slides, or for the online students, you'll use images of those slides, and identify the respiratory epithelium, cilia, goblet cells, hyaline cartilage, basal bodies, basal cells, basement membrane, and fibroblasts. Again, some examples here. Your epithelium is going to look like this. This is what lines most of the conducting portion of the respiratory tract. So that's from the nasal cavity through the trachea and the bronchi. And this tissue is pseudostratified ciliated columnar epithelium with goblet cells. So here you can see the goblet cells here You also have the cilia up here on the top, followed by the basal bodies. These little purple guys are lymphocytes, okay? Then your basement membrane goes underneath all of that with the goblet cells. You can see some blood vessels, and you can tell by how those vessels are shaped. You should be able to tell if they are arteries or veins. So even though it appears, this area up here, even though it appears layered, every cell actually touches, every one of these cells actually touches the basement membrane. So that's why they call it pseudostratified. And then the cilia on the surface, they beat together like in a coordinated fashion to move mucus up the mucociliary escalator. That's what they call this coordinated beating of cilia on the epithelial surface. And what that does is it sweeps debris towards the throat to be swallowed or expelled. And then these goblet cells here actually secrete mucus in order to trap inhaled particles like dust or pathogens. So if you can see cilia and goblet cells in the same place, you're definitely looking at respiratory epithelium. And of course, you will also find things like brush cells, which are sensory. If you look at the trachea is going to be another key landmark under the microscope. It is lined with respiratory epithelium, so you'll still see the cilia and goblet cells here. But underneath that is the lamina propria. Again, we've seen this before. This is just a connective tissue layer, followed by the submucosa, which has the mucus glands. And then surrounding that is the hyaline cartilage. That's what this area here is. And it's usually pretty easy to recognize due to its smooth, glassy appearance. And the open end of the cartilage is bridged by the trachealis muscle, made of smooth muscle, allowing some constriction and flexibility. The adventitia is the outermost connective tissue layer anchoring the trachea in place. So that would be this layer here. Look for distinct layering in this order: epithelium, lamina propria, submucosa, cartilage, adventitia. Then in the lung, we are now shifting to the respiratory zone where gas exchange happens. So you'll be looking at things like the alveola, alveolic ducts, these little ducts in between here. You have two key cell types that line the alveola. You have the type 1 pneumocytes, which are simple squamous epithelial cells. And then you have type 2 pneumocytes, which are cuboidal, and they have microvilli. And their main role is to secrete surfactant, which reduces surface tension and prevents alveolar collapse. And they can also regenerate into type 1 cells after injury. These are some closer views of the alveolar ducts and sacs, and you will notice open bubble-like spaces between. These are the alveoli, okay? And this is a blood vessel, and this is a alveolar duct. Alveoli will look like clusters of tiny thin-walled bubbles because that's where the gas exchange happens. And then type 2 cells are going to be rounder and protrude slightly into the alveolar space, while type 1 cells line the walls. So type 2 cells are going to be out here kind of going into the space, where type 1 cells are more in the lining here. They stay close to the boundary between. This is another version. It's a bit zoomed out, showing the duct here, and then all of these are alveoli. Open, you see the open spaces. These are all alveoli. For exercise 6.3, this activity walks you through real clinical maneuvers like measuring tidal volume during normal breathing, and then pushing your limits with inspiratory reserve and expiratory reserve volume efforts. You will also perform a vital capacity test, which captures the full range of air that you can move in a single breath. The system will then calculate your respiratory rate and help you determine minute ventilation, MV, which is a key indicator of how much air you're exchanging per minute. Make sure to observe the spirometry graphs as it updates. It's going to update with each action, and pay attention to the shape and volume changes. This will help you visually link lung volume data to breathing mechanics. And then once you've gathered your data, compare your findings with standard normal values provided in the simulation. Reflect on any deviations. So are your volumes lower than expected? Could this suggest restrictive or obstructive patterns? Use the background content in the simulation to deepen your understanding of how lung physiology ties into pulmonary health. And this This virtual activity is not only like a practice run for performing spirometry, but it's a great way to visualize how different volumes and capacities relate to respiratory conditions like asthma or COPD. Make sure you take your time, follow the instructions step by step, and then use this opportunity to reinforce key concepts behind how we measure lung function in clinical settings. I do want to take a moment to just familiarize you with the site. You will have a lot of background information. Then it will take you step by step how to do the total volume and the forced vital capacity. It's going to talk again about different things that can affect these values. This is a spirometry graph, and I know this is kind of difficult there's a lot happening here. So what I want to do is I want to take a moment to kind of walk you through this table here. This table is going to summarize the major respiratory volumes and capacities commonly measured in an average young adult male. So knowing these values is important for interpreting the spirometry data in the graph that you're going to get when you're doing this exercise. So first, under measurement, we have each lung volume or capacity by its name, like total volume, inspiratory reserve, etc. The typical value here is a reference number based on an average adult male. These values vary based on age, sex, body size, and physical conditioning, but they also serve it's a helpful baseline. And then over here you have the definition column. This is going to be where you have the descriptions of what each measurement actually means. Let's go through a couple of them. So tidal volume, this is the amount of air moved in or out during a normal quiet breath. Inspiratory reserve, this is the volume that you can inhale after a normal inspiration, like when you take a deep breath in before holding it. Expiratory reserve, this is the volume that you can exhale after a normal passive exhale. And residual volume, which is typically about 1200 milliliters. That's the amount of air that remains in the lungs after full exhalation. This is the volume that helps to prevent lung collapse. And then when you're looking at capacities, down here you have vital capacity. This is the maximum amount of air that you can exhale after a maximal inhalation. Essentially, this is the full ventilatory range. Then you have inspiratory capacity, the total amount that you can breathe in after a normal exhalation. This is the tidal volume plus the inspiratory reserve volume. Then functional residual capacity. This is the ERV plus RV. That's the amount of air left in your lungs after a normal exhale. Total lung capacity is the total volume of your lungs when fully inflated. It includes TV, IRV, ERV, and RV. And as you look at this table and at the graph, try to focus on these two things. What does each value represent functionally? Like, what kind of breathing movement does it describe? And then how do the volumes combine to form capacities? Many formulas are simple additions of the component volumes. And then once you understand those things, this is going to help you understand the physiology of what's going on and prepare you for interpreting spirometry results, diagnosing respiratory disorders, and working through clinical case studies. Thank you guys for watching. Don't forget to do the reading. Check the description for more videos related to these topics and leave your questions for me in the comments below.